Hello, and this is chapter 2 in physiological psychology and the textbook Brain and Behavior. So let's take a look at chapter 2. Communication within the nervous system. A philosophical zombie or P-zombie in the philosophy of mind and perception is a hypothetical being that from the outside is indistinguishable from a normal human being who lacks conscious experience, qualia, or sentience. For example, if a philosophical zombie was poked with a sharp object, it would not feel any pain sensation, but could behave exactly as if it does feel pain. It may say, ouch, recoil from the stimulus, and say that it is feeling pain. Question 1, for the class, can we reduce consciousness to the physical parts in the brain? What if an exact physical copy of you, let's say in the future, some Netflix series were made down to every single detail? Would it be you? Would something be missing? That subjective you, what is termed qualia? Some students have said that it would be you, that exact physical copy, but after the copy was made, that copy would have different experiences than you, and so would be different. What do you think of this position? Some questions to ponder for the class. Of course, the P-zombie argument is put forward by the dualists. The dualists say that there's something that's beyond that physical brain, that there's some type of qualia or sentience or soul, if you will, that's beyond that. And the materialistic monists say, no, that conscious experience, qualia, sentience, what it's like to be you, is produced by brain activity. The exact physical copy. I think the problem with the P-zombie argument is that it presupposes that one could make a exact physical copy that also has you in it. In other words, it presupposes that the brain activity does not indicate qualia or conscious experience. In my version, as pretty much materialistic monist, if you will, brain produces conscious experience. The subjective experience of what it's like to be you is particular to your brain activity. Something to think about. Other things to think about is that one looks kind of funny, huh? Right down here with the eyes and the brain stuff. Neurons. Neurons make up the brain, spinal cord, and nerves that connect to our bodies, and they control all function in our bodies. And this reminds me of the bodies exhibit, both of these, where if you ever get a chance to go see those, are pretty cool, where they show the human body without skin and those types of things. And even saw a horse without skin. You can see all the musculature and stuff. Pretty cool. Neurons are specialized cells that convey sensory information to the brain, integrate that information and transduce it into something the brain can use and understand. For example, photons into neural impulses. Carry out operations involved in every thought, feeling, and action that you have. Transmit commands out into the body to control muscles and organs, and there are 86 billion neurons in the human brain alone. Parts of the neuron with function, so take a look at this. Pause the slide if you want. The dendrites receive signals from other cells, the cell body that organizes and keeps the cell functional, cell membrane that protects the cell, axon hillock, very important for action potentials, it generates that action potential impulse in the neuron. The nucleus controls the entire neuron. The axon transfers signals to other cells and organs, while As the action potential goes down the axon, it jumps in a process called saltatory conduction along nodes of Ranvier. Schwann cells or oligodendrocytes produce the myelin sheaths, and you can also, which increases the speed of the signal, sort of makes white matter white. And axon terminals form junctions with other cells and release neurotransmitters that the postsynaptic neuron, the next neuron, will be sort of catching with these dendrites, if you will, as a lock and key phenomenon. Neurons, one way to classify them is by structure, and there's 4 categories based on cell body placement and the number of protrusions coming from the cell body. Unipolar, let's see an example right here. Only one structure extending from the soma. Bipolar, right here. One axon, one dendrite extending from the soma, do not occur regularly, but involved in the senses. Multipolar neurons contain many dendrites, one axon. Pseudo-unipolar, only one structure that extends from the soma, but later branches into 2 distinct structures. You can also classify neurons according to function. 3 main types of neurons with variations within those types: the motor neuron, sensory neuron, and interneuron. Motor neuron function is to carry commands to the muscles and organs. Multipolar structure located throughout the nervous system. You can see the multipolar structure here and attaching on to a muscle fiber. Axon terminals. Sensory neuron function is to carry information from the outside of the world, of course, in the body to the spinal cord and brain. Most sensory neurons are pseudo-unipolar, meaning that they have an axon that branches into 2 extensions, one connected to dendrites that receive sensory information, another transmits this information to the spinal cord. Bipolar neurons, of course, involved in the senses, as previously mentioned. And interneurons function is to connect one neuron to another in the same part of the brain or spinal cord. Most abundant of neurons in your body. Multipolar structure, as you can see here. Located in the brain and spinal cord, has short axon that communicates locally with other neurons close by. So you can see that's a pretty good communicator with neurons close by, as opposed to having a really long extended axon to transmit information in a distance across the body. Other types of neurons: pyramidal, Purkinje, and granule. Sorry, Purkinje, if I hacked your name there. Lipids. So let's take a look at this neural membrane, which is semi-permeable, made of lipid or fat and protein. The lipids, each lipid molecule has a head, as you can see right here, and a tail. You can see right here, the little tails. The heads are attracted to water both outside the cell and inside the cell. And the tails are repelled by water, so they face each other, as you can see right here, making it look like a little fat sandwich or something like that. Creates a double layer membrane that holds the cell together and controls the environment within and around the cell. Waters and gases pass freely through, but impermeable to charged molecules, ions. These must pass through protein channels called ion channels. Let's take a look at those. They're narrow water-filled tunnels that allow only ions of certain size and or charge to pass through. And you can see examples of the protein channels right here in this diagram. Creates a selectively permeable membrane, as you can see, because it only allows certain things to come through, certain ions to come through. Neural membrane, neurons lie in a salty solution that is positively charged compared to inside the cell. The selective permeability of the membrane creates polarization, which means that there's a difference in electrical charge between the inside and outside of the cell. This difference in electrical charge between 2 points is referred to as voltage, similar to what a battery might have with the positive and negative charge. Resting potential, difference in charge between the inside and outside of the membrane of a neuron at rest. Of course, definition of it. It is between -40 and 80 millivolts in different neurons. A typical neuron's resting potential is around -70 millivolts. Resting potential is caused by unequal distribution of ions on either side of the membrane. At rest, more positive charged ions are on the outside compared to the inside. Outside the membrane is mostly salt, sodium, and chloride. So sodium is the Na+, chloride, the Ci- ions. Inside the membrane contain mostly potassium, positive ions, and organic ions, which are negative. You can see there positive and negative signs. Take a look at a brief video that talks about resting potential. I'm going to pause right here in the video and come back for the next slide. Here we go. Force of diffusion. Ions flow from high to low concentration. They want to be equally concentrated. So basically, these are two sort of forces that you learn about in chemistry. One is, of course, what I just listed, the force of diffusion, where ions really want to flow from high to low concentration. That's the way nature is. They want to be equally concentrated. Nature likes balance. The other part of nature and molecules of chemistry, a law of chemistry, is electrostatic pressure, and that's that opposites attract. Ions attracted to the opposite charge and repelled by the same charge. So of course, positive is attracted to negative, vice versa. Positive is repelled by positive. Both of the above forces should move the organic anions, which are inside, out. Diffusion looking to move from a higher to lower concentration, and electrostatic pressure attracted by the outside positive charge. These anions are too large to pass through the ion channels outside the cell. Chlorine, which is outside the cell, is repelled by the anions. The forces of electrostatic pressure mainly keeping them from going inside. So essentially, chloride and the outside and the anions on the negative side, they cancel each other out and they do not become players in this possible process of charging and sending everything from your thoughts to your actions to literally hearing and seeing this lecture right now, right? Just neuron activity. So what enables that electricity? Sodium and potassium become the real players in this. Potassium moves easily across the ion channels, and slight tendency for potassium to move out as forces of diffusion are stronger than the electrostatic pressure. But as they move out, some are pulled back by the anions. Sodium moves less freely across the ion channels. In resting state, some ions trickle through the ion channels. However, the sodium-potassium pump keeps the balance of that negative resting state on the inside. And it does this by moving 3 molecules of sodium out for every 2 potassium moves inside. Sodium ions. Pump also rebalances neurons' negative resting potential by pumping sodium out and potassium back in. And so this is just a picture of that and diagram and discussion of that. So you can take a pause and look at that. That's something from the text, as well as take a look at action potential and ion channels by a guy who talks really fast. That's a good video. Okay, now remember there are 3 different types of ion channels. There are ligand-gated ion channels, and these are chemically gated channels opened by ligands. These are neurotransmitters or hormones. There are voltage gated ion channels, and this is what we were just talking about, electrically gated channels opened by a change in the electrical potential of the membrane. Remember the sodium rushing in, potassium going out. And mechanically gated ion channels opened by physical distortion of the cell membrane, touch controlled by these channels. So what's a local potential? And this is basically in preparation for talking about action potential. And of course, these are things that cause you to think, touch, experience life. Local potential, a neuron is usually stimulated by inputs that arrive on the neuron's dendrites and or cell body from another neuron or from a sensory receptor. This effect may be excitatory or inhibitory, depending on the ligand, and the characteristics of the receptors. Sodium, potassium, chloride ligand-gated ion channels, either open or closed, for example. Excitatory signal causes a slight partial depolarization in that area of the membrane, whereas an inhibitory causes a slight hyperpolarization. These can cancel each other out. So that's basically how things are sort of summed up and sort of kept things in balance and checks and balances, right? You have some excitatory, but you also have some inhibitory. So even though the neural impulse, the action potential, is just at one speed, there's different frequencies that the neuron can fire. And then as you can see, there's different inputs that the neuron can have, both excitatory and inhibitory, making it its functionality much more complex, and its abilities much more varied. If excitatory, the sodium flows in and across the inner part of the membrane, causing a partial depolarization, less than 1 millivolt, but the electric current is decremental, so it dies out, for example, and so it is called a local potential. And you can see right here some of the local potentials that did not, A and B, that did not reach the full area where there can be an actual action potential, so it didn't go above that 10 millivolt threshold. For an action potential to occur, there must be many local potentials that occur across the membrane of the neuron. So as you can see here, a bunch of local potentials are happening, and the summation will occur at the axon hillock in the trigger zone. It's what's happening there is it's algebraically summed up. If depolarization reaches threshold of excitation, it increases by 10 millivolts from negative 70 millivolts, for example, to 60 millivolts, the action potential will be triggered. On average, the resting potential is at negative 70 millivolts. What is an action potential? It's a rapid depolarization and slight reversal of the usual membrane polarization. Occurs when depolarization meets or goes beyond the threshold of excitation. So you can see, and of course, just what I said before, it increases by 10 millivolts. So you can see right here, there were a couple local potentials that were failed initiations, but let's say, for example, or those show them, and on one time period or experiment, another failed initiation. They did something that actually met that threshold, touch, oh, I could feel that touch or something like that. And causing a depolarization, getting to that set from 70 to 60. And as you can see right here, once it hits that, it goes way up to plus 40. What happens, summation, the multiple local potentials are summed up, remember, at the axon hillock. And a process like I was talking about with depolarization, we actually have an action potential firing down the axon. Depolarization with the potential across the membrane reaches a threshold. Remember we were talking about here voltage activated ion gates. Those are sodium ion gates. Will open, sodium ions enter, and this causes the membrane potential to depolarize past zero to a reversed polarity. Negative 70 millivolts becomes plus 50 millivolts at its highest amplitude of the action potential. So it goes from a negative to a positive charge. So what happens then? You can see the process. It goes to a positive charge, and then after that, there's a process of repolarization. That's when the action potential reaches its peak. Voltage activated sodium gates close, but potassium ions flow outside of the membrane due to their high concentration inside the neurons as opposed to outside. So then you have this falling down of the millivoltage, and it falls below the regular threshold. And what's that called? That's called a hyperpolarization. As you can see, it's temporal hyperpolarization occurs before the membrane returns to its normal resting potential. This is due to the potassium gates opening wider than usual, allowing potassium to continue to exit past the resting potential. So it overshoots it, and then it comes back to the resting state. After the action potential, the neuron has more sodium and fewer potassium ions inside for a short period. And so you have that hyperpolarization right there. Again, beyond the hyperpolarization, and that's adjusted by the sodium potassium pump to get back to the resting state. And that's shown right there. And this is from your textbook, same thing that we just talked about. What is myelin? Many axons are myelinated. See right here, the axon. I think it's white matter, its name, as I mentioned earlier. It's fatty tissue that wraps around the axon like a jelly roll, provides support for the axon, and also insulates it, which allows electrical messages to travel faster, which saves energy, lots of important things. Does not allow ion channels to form on it, so sodium and potassium do not escape, and the current travels down the axon by what's called a local potential. See right here, so local potential, nodes of Rambier, local potential, or down the axonal section, nodes of Rambier. What are the nodes of Rambier? They are myelin gaps on the axon, as you can see right here, where ion channels are allowed to form. Gaps are perfect distance apart, placed just in time for the local potential to not diminish, where the current gets another burst with the ion channels opening and closing, and the propagation of the action potential. This charge, as it goes down, allows the electrical charge, if you will, to go like this and sort of jump. And the process of it jumping is called saltatory conduction. The charge travels fast in a myelinated section of an axon as if it jumps from node to node. So the charge will come down right here. And then, as you know from the discussion, that you will have ion channels in these nodes of Rambier that will then give it another boost, if you will, another potential local potential fire or action potential firing that will then make that local potential travel down that next segment. So what happens is it ends up sort of jumping really fast and getting sort of a new charge each one. A new burst, if you will, in each nodes of Rambier with the salt infusion and potassium going out. Occurs only on the axon. Dendrites use chemically gated ion channels, ligands such as neurotransmitters and hormones open and close the ion gates, whereas axons use voltage gated ion channels, instigated by changes in voltage. This change, the action potential, follows an all or none law. It occurs at full strength or not at all. and the action potential is non-decremental. Message travels over long distances at the same amplitude. The domino effect is something where depolarization that occurs during the action potential triggers nearby sodium channels to open as well. Thus, a new action potential is triggered right next to the first one. That action potential in turn triggers another farther along, creating a chain reaction of action potentials that move through the axon. Thus, the signal flows from one end of the neuron to the other. Nothing physically moves down the axon. Instead of a series of events occur in succession along the axon's length, much as a line of dominoes standing on the end knock each other down when you tip the first one. What's the absolute refractory period as well as a relative refractory period? Well, similar to sex, if you will, it's a period of time during which a second action potential absolutely cannot be initiated. No matter how large the applied stimulus is, the sodium channel, ion channels are inactivated in this case. And there's also the relative refractory period, and it's just as it sounds, the interval immediately following the absolute refractory period during which initiation of a second action potential is inhibited, but not impossible. During this time, the potassium channels remain open and make the neurons slightly more negative than usual, which means that only a stronger than threshold stimulus can make the neuron fire again. With increasingly stronger stimuli, the neuron will fire earlier and at a higher rate. Thus, the axon encodes stimulus intensity not in the size of its action potential, but in its firing rate. And this effect is called the rate law. Neurotoxins can affect ion channels involved in the action potential. Tetrodotoxin from the pufferfish blocks sodium channels and causes paralysis in respiratory muscles, leading to death. Snake venom blocks sodium, potassium, or calcium channels, and the scorpion venom opens sodium channels prolonging the action potential. Beneficial drugs affect these ion channels as well. Local anesthetics block sodium channels, and general anesthetics work by opening potassium channels. So you can see how important these ion channels are for our very survival, our consciousness. Consciousness is not determined by the soul or the sentience, that subjective view, if you will, that's outside the brain. I asked my daughter, who's in 3rd grade, and we're talking about the brain. Oh, what do you think about your brain? Is that like where you think, where that helps you to think, feel different things? She's like, yeah. I'm like, okay, so you would identify you, like your experience of being aware and all that is coming from your brain? She's like, yeah. She's like, where is it supposed to come from? Your ear? Your toe? Good point, kid. Good point, kid. She's still in the concrete thinking stage, though, so you can hit her up when abstract thinking comes along and see if she still agrees. Glial cells, a Greek word for glue, provide a number of supporting functions for neurons. You can see the types of neuroglia. Go over a couple of them. See how they're supporting various neurons connecting to them. Microglia are the brain's immune cells. They serving to protect against injury and disease, removes the toxic agents, and clears away the dead cells. Synaptic pruning in child's developing brain, it's responsible for that, eating at synapses. And the situation may be different with the neurodegenerative disorders such as Alzheimer's, where the microglia are hyperactivated, promoting neuroinflammation that can lead to the characteristic toxic protein deposits seen in Alzheimer's, the amyloid plaques and their neurofibrillary tangles. Nuclear, having trouble with that one. Not gonna do it. Oh wait, that's the wrong one. Gutnista, not a fashionista. Moat my eye. Astrocytes, mostly a star shape, provide nutrients, influences synaptic activity, recycles glutamate, and releases it. For example, repair a nervous tissue following injury, facilitate, regulate neurotransmission, and support the blood-brain. Oligodendrocytes are responsible for myelin. Can I say that fast 5 times? Myelinating neurons in the central nervous system, and the Schwann cells are responsible for myelinating neurons in the peripheral nervous system. Other glial functions, scaffolds for migrating neurons, providing energy to neurons, and neural connections. And remember, we're all about connections and connectivity, especially in the brain. There's 7 times more neural connections when glia are present, shown in the slide down below with cultured neurons. Grow neurons in a dish. Synapse, Latin to grasp, it's a connection between 2 neurons. So it's the gap between the presynaptic neuron, which is the one that transmits the signal, that's just a fancy word for that, and the postsynaptic neuron, the neuron that receives the signal. The synaptic cleft is the gap between the 2 neurons where the neural communication takes place. Here's a video on synapses and how neurons communicate with one another, if you want to take a look at that. Video. So some more information about synapses, vesicles, the axon terminal. There are membrane enclosed containers called vesicles, which store the neurotransmitters. So this is just an example of one of the terminals on the neuron right here, as you can see. Exocytosis is a process where action potential causes calcium ions to enter the terminals from the extracellular fluid. Calcium ions cause the vesicles clustered nearest the membrane to fuse with the membrane, causing it to open, where the neurotransmitters spill out into the synapse. So that's what's going down. You got the action potential, and then that causes the calcium to enter into the terminal, bind with those vesicles and cause the vesicles to spill out their neurotransmitters. What happens when those neurotransmitters spill out into the synaptic cleft? Well, it's what's called a lock and key phenomenon. On the postsynaptic neuron, the neurotransmitter docks with specialized protein receptors that match the molecular shape of the transmitter molecules, like a lock and key, which in turn open ion gates in the neural membrane. 2 types of protein receptors on the postsynaptic neuron. They're ionotropic and metabotropic receptors. Ionotropic receptors, of course, open ion channels directly to produce immediate reactions required for muscle activity quickly and sensory processing. Metabotropic receptors open ion channels indirectly, slowly, to produce longer lasting effects. Opening of channels sets off the graded potential. Synaptic transmission is much slower, of course, than axonal electrical transmission. Activation of receptors on the postsynaptic side, the one that's the neurons that receiving that information, has 2 possible effects on the membrane potential. Hypopolarization creates an excitatory postsynaptic potential. Hypopolarization, same as depolarization, right? We talked about earlier, creating that action potential, right? An EPSP, that's what we'll say from now on, opens sodium channels. An excitatory postsynaptic potential opens the sodium channels. And this makes the postsynaptic neuron more likely to fire an action potential, or the more graded potentials, which equal the action potential. Hyperpolarization creates inhibitory postsynaptic potential, IPSPs. An IPSP opens potassium or chloride channels, or both, and that would mean that the neuron is less likely to fire an action potential. Well, what's the value of having IPSP? Remember, it's all about complexity and messaging, and so it increases the complexity of messaging as input from one source. Potentially can partially or completely negate input from another source. It also prevents runaway excitation, one cause of uncontrolled neural storms that sweep across the brain during an epileptic seizure. There's a deficiency in receptors for the inhibitory transmitter GABA. So little checks and balances. EPSPs and IPSPs are graded local potential. The potential spreads down the dendrites, across the cell body to the axon hillock. If the potential moves to threshold, action potential will occur, as we talked about earlier. A typical neuron receives input from 1,000 other neurons, giving it input to the dendrites and onto the cell itself, cell body. Temporal summation describes where in this summation at the axon hillock, it combines the potentials arriving in a short time apart from either the same or separate inputs, and spatial summation describes how it combines inputs occurring simultaneously from different locations on dendrites and the cell body. The neuron acts as an information integrator, in that it is summating the information, and a decision maker. Excitatory and inhibitory inputs combine algebraically and fires when above the threshold. They're ready to fire. Those neurons are ready to fire, right? And you can see right here an example of what we were just talking about. Point 1 is going to be an EPSP. That doesn't fire an action potential. Temporal summation of 2 EPSPs right here gives it a little bigger of a firing, and temporal summation of 3 EPSPs reaches threshold. Spatial summation of EPSPs reaches threshold. An IPSP, and then the hyperpolarization, and then temporal summation of 2 IPSPs. Neurotransmitters must be removed to allow neurons to fire frequently and prevent neurotransmitters from spilling into nearby synapses. Reuptake is one process whereby the neurotransmitter brought back into the terminals by the membrane proteins called transporters. So basically that's one way to get rid of the neurotransmitters in the cleft or spilling into nearby synapses. And inactivation, enzymes will break down the transmitter in the cleft as well. Drugs, SSRIs, selective serotonin reuptake inhibitors, or various drugs that limit the enzymes, are ways in which you can affect those neurotransmitters and keep them in the synapse, if you will. Agonists are drugs that mimic natural transmitters and stimulate receptors in cells. Opioid drugs such as heroin and morphine act as agonists, stimulating receptors for opiate-like transmitters in the body. Whereas antagonists are drugs that block neurotransmitter receptors. Naloxone and naltrexone are antagonists which occupy the receptor sites without activating them. And these are, of course, treatments for opioid overdose. And plant toxin curare blocks nicotinic acetylcholine receptors at the muscle, causing paralysis. Some drugs enhance or reduce transmitter effects. For example, as I mentioned earlier, SSRIs are selective serotonin reuptake inhibitors that block the reuptake of serotonin and keep serotonin in synapses. And some prevent neurotransmitter inactivation. Monoamine oxidase inhibitors, or MAOIs, inhibit enzymes called monoamine oxidase, which typically breaks down monoamines such as serotonin, norepinephrine, and dopamine. So it inhibits the enzymes and thus keeps them in the synapses. Regulating synaptic activity is important so the system can function effectively. The activity doesn't drift out of control or fade away. Take a look at this really important slide about ways that regulation is affected. In one right here, you can see the terminal from another neuron forms an axoaxonic synapse and adjusts the amount of neurotransmitter output. So that's one way to control it. Autoreceptors also sense transmitter amount and can adjust output. And receptors can even change in number and sensitivity as well. So there's various ways to regulate the synaptic activity. In addition, glial cells surround the synapse and prevent the transmitter from spreading to other synapses, and they also absorb and recycle the transmitter for neurons use, and sometimes even produce transmitters, such as releasing glutamate to regulate the presynaptic transmitter release or directly stimulate postsynaptic neuron to excite it or inhibit it. These are the astrocytes, of course. Neurons can release also more than one chemical, which contradicts Dale's principle. It can be a fast-acting one and a slow-acting one, or more. Be two or more fast-acting neurotransmitters or neuromodulators that influence the neurotransmitters, excitatory and inhibitory transmitters at different synapses, and different subtypes of receptors as well. And talking about neural co-release and co-transmission, take a look at this slide. In co-release, the neuron can limit the exit of larger chemical messengers by partially opening the fusion pore. You can see in A. The messages are in different vesicles in the same terminal. The vesicles different differential calcium sensitivity provides release selectivity. And packaging the transmitters in separate terminals allows the neurons to have different effects at terminal destinations. So you can see the multiple ways, as we've been going over, that neurons can modulate the way that they communicate to one another, such that it can become much more complex. Take a look at this slide for a moment. I think it's really important because you can see how these various neurotransmitters can affect different parts of ourselves and every waking or even sleeping parts of our lives. Take a look at serotonin, for example. It's involved in mood, sleep and arousals, aggression, depression, OCD, and alcoholism. Acetylcholine, transmitter of muscles and brain, involved in learning. Well, it's glutamate, also involved in schizophrenia. GABA, involved in alcohol response, as well as tranquilizers such as benzos, etc. Epinephrine, norepinephrine, dopamine. It's our reward neurotransmitter. Contributes to movement control, promotes reinforcing effects of food, sex, and abuse drugs. We wouldn't want to eat, or have sex if we didn't have dopamine. Dopamine is the neurotransmitter that makes us feel good about those things. In fact, you will have dopamine release by just looking at yummy food. Why am I talking about this at lunchtime? Well, hopefully slides are almost over. I can fulfill that dopamine urge. And a couple other some neuropeptides. Coding of neural messages. Neural impulses vary in intervals, with bursts separated by different intervals. Does this temporal variation in firing form a code that the brain can use? The answer is yes. The brain also has specific labeled lines specialized for different types of stimuli for each sense. See the effects of coding of neural messages. And remember that while there is significant localization, prefrontal cortex, for example, has specific functionality, occipital lobe, Broca's area, Wernicke's area. It's also important to know, as the connectome shows, which is the picture down here, the brain relies on interconnectivity heavily as well. Neural networks add further complexity in neural processing. Groups of neurons that function together to carry out a process. That's what the neural networks are. The Human Connectome Project is a large-scale multi-university effort to map the brain circuits. Use a white matter imaging technique called diffusion tensor imaging. And the colors, as you can see down below, represent varying strengths of connection, and you can see how the brain is immensely connected in different parts across brain, bottom to the top. And that is the end of chapter 2.